in 20 days of amino acids, the amino acid phenylalanine, which is a great segue into talking about a condition called PKU, phenylketonuria, which is a disease in which patients can't break down phenylalanine, they can't convert it into tyrosine, and therefore they can't make all the things that they need to make with tyrosine, and instead they build up some byproducts that have distinctive smells and react with things to make colors that then allowed this condition to be discovered. And now that this condition is discovered, we have early tests for it, as well as some treatments. And so I'm going to tell you about these various things and the answer to why you have those warnings on your gum and stuff that say phenylketonurics contains phenylalanine. Because yes, things like aspartate are dipeptides that have phenylalanine in them, and therefore even that sugar-free gum can actually cause problems to people with PKU. So let's dive in. The basic idea is that the amino acid phenylalanine has this benzyl side chain. And if we think about just the ring portion, this benzene ring, if something just has this benzene ring, then we call it a phenyl group. And so if you look at this, if we took off the ring, we would have alanine. And so with the ring, we have phenylalanine. So although the ring with an extra carbon is a benzyl group, the ring itself is a phenyl group. And this will come into play when we think about the names of some of the byproducts. Let's start with big picture. Patients with phenylketonuria have a mutation in an enzyme called phenylalanine hydroxylase. And phenylalanine hydroxylase, what its role is to convert phenylalanine into tyrosine. So about half of the phenylalanine that you get in your diet is actually used to convert to tyrosine. And I say that you get in your diet because phenylalanine is one of those essential amino acids, meaning we can't just make it ourselves, and instead we need to get it through our diet. So what phenylalanine hydroxylase does is it actually adds this hydroxyl group, this alcohol group, this OH, onto phenylalanine, and that gives you tyrosine. People can have mutations in this phenylalanine hydroxylase enzyme, often abbreviated PAH, and then they can't do that conversion to tyrosine. And this is important because tyrosine is typically used not just to make proteins, but also to make hormones, to make melanin, and it can get broken down for energy and spare parts. If you can't do that conversion from phenylalanine to tyrosine, then that phenylalanine can't get broken up the normal way. It builds up alternative metabolites, including phenylketones, which gives it the name phenylketonuria because these ketones can build up in the urine. And it also causes problems with having less of those things that were made from tyrosine. It also throws off energy production when you're going that alternative pathway. And it's going to make it harder to get all of the amino acids into your brain. Well, not all of them, but a number of them. The large neutral amino acids, these are sharing a common transporters with phenylalanine. And so if you have a ton of phenylalanine building up, then what's going to happen is those other amino acids are going to have a harder time getting in. So there's still a lot of work into trying to figure out what exactly causes all of the symptoms from PKU. It primarily causes developmental delays and neurological difficulties. However, most of the enzyme is actually found in your liver, so it's not exactly clear, and there's still a lot of research being done on this. In a little more detail, those hormones that you can get from tyrosine include things like dopamine and adrenaline, noradrenaline, as well as thyroid hormones and melanin. So like skin pigmentation, you can have problems with skin pigmentation, neurological problems, thyroid problems, all sorts of problems that can come if you can't convert phenylalanine to tyrosine, problems that come from the lack of the tyrosine, as well as problems that come from the process of making phenylalanine get broken down a different way. And so normally what would happen if you had a lot of phenylalanine was you convert it to tyrosine, and then you could break down that tyrosine to get fumarate and acetoacetyl-CoA. And those were therefore going to be able to use to make ketone bodies or be able to use to go in through the citric acid cycle, which could be used to make glucose. And so we call phenylalanine and tyrosine glucogenic and ketogenic. That's if things happen the normal way. Now let's go and think about what happens when you have a lot of phenylalanine and you can't go that normal breakdown route. Instead, you still want to get rid of all that excess phenylalanine. And so you're going to do some different breakdowns. One of them is you need to remove the amino group. So similarly to all amino acid catabolism, we've got to remove that amino group safely, typically through like the urea cycle, and so that we don't just build up ammonia. The nitrogen groups from phenylalanine can get transferred onto alpha-ketoglutarate to give you glutamate, similarly to how we can pass the amino groups from any amino acid onto alpha-ketoglutarate to give us glutamate. And this glutamate can be used in the urea cycle to get rid of the nitrogen group as ammonia. And what you're left with is phenylpyruvic acid. So remember that if we think about just this benzene ring and attached to something, we call that a phenyl group. And so if we were to see, okay, we've got these 3 carbons and we've got this acid group here and this ketone group here, well, what we're looking at here is pyruvate. And so if you put a phenyl group onto pyruvate, you get phenylpyruvic acid. This phenylpyruvic acid, keep it in mind because it's going to be one of the clues that help them discover this condition. Alpha-ketoglutarate is normally in high demand, and so not only does it need it for the breakdown of other amino acids, but it's also needed in the citric acid cycle. And so if you are using all of it for your phenylalanine breakdown, well, now you're going to be diverting from those processes, and you can cause the energy balance to get all out of whack. Phenylpyruvic acid can also get further modified, such as getting oxidatively decarboxylated, which is going to give you phenylacetic acid. And this phenylacetic acid, this is going to be the key to finding this condition. So let's get into how they found it. So this phenylacetic acid, it makes pee smell. It gives it a musty smell. The smelliness of this phenylacetic acid provided the key clues to what was going on in these patients, Liv and Dag, who were the young children of Borgny and Harry Egeland, a couple living in Norway in the 1930s, and Liv and Dag had severe developmental delays, and their urine had a musty odor. They had family connections to a doctor named Dr. Asborn Folling, and they took the urine to this doctor and was like, can you help us figure out what's going wrong in our children? Now, Folling, he noticed that musty smell, and he thought, well, it's kind of like the smell that you get in diabetic patients. And the urine of diabetic patients also often has like a musty, acetone, fruity-ish type odor if it's untreated. And so he put this urine through one of the tests that you can do with diabetic patients to detect diabetes. This test involves adding a chemical called ferric chloride. And when you add that ferric chloride to urine of patients with diabetes, typically it'll turn purpley. However, when he added the ferric chloride to Liv's urine, it turned dark green. And he did not know what was going on. And so he kept having the parents bring him fresh urine. Ultimately, what he did was he took a total of 20 liters of urine and he purified out a product from it and did a bunch of chemical tests on it and determined that product was phenylpyruvic acid. These findings were published in German in 1934, and he suggested the name phenylpyruvic oligophrenia. It's called Folling's disease in Norway, and then it got the name PhenylKetonUria from another doctor, Lionel Penrose, because of the phenylpyruvic acid detected in the urine. Just a note that these days, phenylalanine itself is measured in the blood using mass spectrometry, often abbreviated mass spec, which is a technique which breaks down molecules into charge fragments and then measures their charge to mass ratio to get a sort of fingerprint they can then compare to the known fingerprints from molecules. We'll get back to this idea of measuring phenylalanine and phenylalanine byproducts because it's important to detect the condition early and treat it. And we'll also talk about treatments. But back to the history. He had found that these patients had this phenylpyruvic acid. So initially he found it in Liv's urine, and then he found it in Dag's, and then he did some other research and found that other patients with similar symptoms had this same buildup. Where was that coming from, however? This work was happening in Norway in the 1930s, and now we're going to switch to the USA in the 1940s and the lab of George Jervis, who was working in the research department of the New York State Department of Mental Hygiene. George Jervis fed both healthy patients and patients that had this PKU condition phenylalanine. In healthy people, phenylalanine gets converted to tyrosine. And so if you feed them phenylalanine and then do a test to detect tyrosine, what you're going to see is that the tyrosine levels increase. So you can see that he tested a number of different people. And you can see fasting, if you have a level of about 1, and then you feed them phenylalanine, the levels of tyrosine increase as that phenylalanine gets converted to tyrosine, and eventually then it starts to go back down to normal. But what about patients with PKU? When he fed patients with PKU phenylalanine, he saw that their tyrosine levels remained constant, signaling that the problem was with hydroxylating phenylalanine to make tyrosine. There was a holdup in the conversion of phenylalanine to tyrosine, and therefore the levels of phenylalanine were remaining high, but the levels of tyrosine weren't increasing. Now that we know the cause, or at least we have an idea of the cause, how can we actually go and treat it? The most obvious kind of treatment was, okay, well, let's just not give them phenylalanine. And so this is what scientists did. They took protein and they took advantage of the hydrophobic nature of phenylalanine. So hydrophobicity, this is basically how much water hates you. And so if water hates you, then if you give the molecule a choice between hanging out with water and hanging out with something else hydrophobic, well, the water is going to exclude you, and so you'll end up hanging out with the hydrophobic thing. One hydrophobic thing that is often used to separate hydrophobic things from hydrophilic things is charcoal. So basically carbon. So if you take a mixture of proteins and you mix it with this activated charcoal, the phenylalanine is going to stick to the charcoal, whereas the other amino acids won't. And therefore, you're able to get a diet that does not have that phenylalanine, but it has the other amino acids. So what Dr. Horst Bickel, Eli Wolf, and Evelyn Hickmans did was they went and they treated a 17-month-old PKU patient with a diet made in this way. This was happening in 1953. They took this patient and fed her a no phenylalanine diet and measured a few different things. One is the measure of their plasma phenylalanine, so how much phenylalanine is in their blood. They saw that when they didn't give the patients phenylalanine, the levels of phenylalanine decreased. However, so did their weight, as well as the mental status. And so it appeared that the patients needed some phenylalanine. So when they added a little bit back, things got much better. And then when they briefly added a lot to test, the phenylalanine level spiked and the neurological condition quickly deteriorated. So this experiment showed that you needed a little bit of phenylalanine and that you had limited the phenylalanine. That was going to be beneficial, but you needed a little because phenylalanine is essential. We do need it to make this tyrosine too. This was the basis of an initial treatment for PKU in 1972, a protein formula for PKU, so you can think of like a protein smoothie type thing, became the first medical food in the US called Metabolic. And since then, there have been further improvements in treatment. And I'm going to tell you about a couple of these, but in order to kind of understand what's going on, we need to first think about what kind of mutations can actually cause PKU. PKU causing mutations are typically what we refer to as autosomal recessive, meaning that you need two faulty copies of the gene, so like one from each genetic parent, and these are located on autosomes, so basically not your sex chromosomes. Patients are often compound heterozygotes, meaning that they have two different faulty versions. So they have two faulty versions, so they do have the condition. However, those faulty versions are faulty for different reasons. And so there are actually hundreds of different PKU causing mutations. They can affect things like the catalytic function, so how well the enzyme can actually do that conversion of the phenylalanine to the tyrosine, how well they can bind a cofactor, so a needed helper molecule called BH4, how much of the protein gets produced, various things like that. So it complicates things, and it also makes it so that treatments need to be kind of more specialized. So let's talk about some treatment strategies. If patients have problems with the BH4, like binding or things like this, if you give them more of the BH4 protein, that can help improve things. And so this drug Kuvan helps patients use kind of the normal breakdown pathway of phenylalanine by giving them some of that extra cofactor, this tetrahydrobiopterin, that's needed to do this conversion. And so if they're not very good at using the BH4, but you give them a lot of the BH4, and a stable version of it, well, now these patients might have a better chance of being able to convert more of that phenylalanine into tyrosine. However, if the patients don't have that PAH activity still left over, if the problem isn't just that they're weak, but the problem is actually that they're not making the protein, or they're really have no catalytic activity, they can't do that conversion even if you give them BH4, this strategy isn't going to work. Thankfully, another strategy came about in about 2018, Palisnik. This provides an alternative pathway. So it gives patients an enzyme that we don't have ourselves. We don't have it in our body, but some bacteria have it in some plants. It's called phenylalanine ammonia lydase. And what it does is it takes this phenylalanine, and instead of going and hydroxylating it, it actually introduces a double bond here and removes the amino group. And this gives you a molecule called tranexamic acid, which is able to get metabolized by the liver. Voila. And it doesn't use up your alpha ketoglutarate. This is an example of enzyme substitution therapy. Initially, it was kind of given as an oral treatment, but that didn't work and it was getting digested and not working, but now it's given as an under the skin injection and a stabilized version of it. So it's like pegylated, it's got some modifications. These treatments can be life altering. However, they need to be started very early. Early diagnosis and treatment are critical, as seen in this picture of an untreated boy versus his younger sister, who both of these have the same genetic condition, but the sister's condition was taught early in infancy and promptly treated, whereas her brother was not. PKU is actually the first metabolic disorder to be included in newborn screening, and it's mandatory in the United States. The tests for it are often referred to as Guthrie tests, after the original screening test I'll tell you about, and that was developed by Robert Guthrie, who is a physician, microbiologist, and father of a child with PKU. That wasn't the first test, however. So remember that in patients with PKU, they have a buildup of these byproducts, including phenylpyruvic acid in their urine, as well as a buildup of phenylalanine. So, compared to a normal person who might have 1 to 3 mg per 100 mils of phenylalanine in their blood, patients with PKU might have 20 to 60 milligrams per 100 milliliters. So very high levels. If we can detect phenylalanine, or if we can detect one of these byproducts like phenylpyruvic acid, then we would be able to detect the potential presence of PKU. The first test took advantage of that whole ferric chloride turns this urine green thing. And so they did urine tests. This was first widely used in California in the 1950s. And they could use like a dropper to put ferric chloride onto a urine-coated diaper or a dipstick that was coated with ferric chloride, and therefore they could look and see if it turned to green. However, that was detecting this phenylpyruvic acid, which doesn't build up until a few weeks after birth. And so they wanted something that could be even quicker, because the earlier you start the treatment, the better. This led to blood-based tests, and these started being used widely in the 1960s and are referred to as Guthrie tests. Now, the original Guthrie test was a bacterial inhibition assay. And what they did was they took bacteria, Bacillus subtilis, and they grew it on this special food that instead of having phenylalanine, it had this kind of competitor. And the bacteria would die unless you gave them a lot of phenylalanine. And so what they did was they were able to put little spots of blood. If the blood has a ton of phenylalanine because these patients have PKU, then that's going to be enough phenylalanine for the bacteria to grow around the spot of blood in these sorts of halos. And this allowed them to then see if there was a lot of phenylalanine in the blood. These days, they just use mass spectrometry. So they can measure the levels of different metabolites, so different small molecules like phenylalanine in the blood. They can also then test for a variety of different conditions. So not just measure phenylalanine, but also measure a bunch of other metabolites, a bunch of other amino acids and things that are commonly out of skew in people who have metabolic conditions, and therefore you're able to screen for a wider range of conditions with a simple blood test. Mandatory testing wasn't the only governmental action. Instead, governments started requiring food makers to put those labels on food that like PhenylKetonurics contains phenylalanine. And this is typically put in places where you might not expect it to be, such as on your sugar-free gum. This is because things like the sweetener aspartame, well, it's actually a dipeptide of aspartic acid and phenylalanine. If you break it down, you then get phenylalanine and aspartate. And you might be wondering, well, isn't that then not 0 calories? Well, it's not 0 calories, but it's a lot sweeter than sugar. And so you lose a lot less. And so it's typically considered calorie free. But when you go and you look and see things like might say like per serving 0 calories, per container or whatever, like 5, that's because often these sweeteners, you still have some calories from them, but they're so much sweeter that you use so much less. And these warnings help give people a heads up that there is phenylalanine in these products, or at least the precursor to phenylalanine in these products. Another kind of technical note is that in order to break down the phenylalanine, you need working PAH, so that enzyme that's kind of associated with PKU, as well as working BH4 recycling, so that cofactor. And a problem with either of those can lead you to have elevated levels of phenylalanine. And so this is referred to as hyperphenylalaninemia, or HPA. PKU is specifically when you have defects in BAH that are causing this HPA, but you can also get it from defects in other aspects of the pathways, which this is then referred to as non-PKU HPAs. Just a technical note. So hope that helps you appreciate that phenylalanine. It's not just for your proteins, it's for a lot of other things, and many of those other things are actually coming from tyrosine that comes from phenylalanine. And in order to do that conversion, you need the enzyme phenylalanine hydroxylase. And with patients with PKU, this is going to be mutated, so they can't do that. That causes all these problems. But if caught early, this condition is treatable. And so thanks to the scientists who worked this out, and hope that you don't have to think about this because you can convert things normally. But if you can't, now you know the science of it.